Hi everyone. So today we're going to learn about a protein called collagen. So collagen is the major structural protein which is present in the extracellular matrix, and also it is one of the most abundant protein which is found in vertebrates. So let us start with the structure of collagen. So a collagen is actually a fibrous structure. That means, it looks like a long thread kind of structure, and then it is rod in shaped. When we studied the structure of hemoglobin, it was a globular protein, right? Because it was present in the form of a globule. But then this structure is present in the form of a rod shaped, right? And it is usually present in our skin, bone, tendon, cartilage, and then teeth. Therefore, it is present in so many places, so it is also one of the most abundant protein found in vertebrates. Now, the length of the collagen, fiber is usually around 3,000 angstrom, and then the diameter of the collagen thread is usually of 15 angstrom. So I've just shown you how does a collagen fiber look like. So this is actually just a small structural unit of collagen, which is known as tropocollagen, right? So this is tropocollagen, and many tropocollagen will cross-link with one another, and then they will finally form a collagen fiber. So we're going to learn about it, don't worry. Now, as I told you, the length is almost 3000 angstrom, and then the diameter is around 15 angstrom. And then, in one polypeptide chain, as you can see over here, there are three polypeptide chain, right? One is in the blue, one is in the green, and the other one is in the pink. So we have three polypeptide chain, and each polypeptide chain is made up of 1000 residues of amino acid. Now, the configuration that we're going to learn about, the collagen molecule, is usually left-handed configuration. That means, it will go anti-clockwise direction. But then, when all these three fibers come together and then they coil with one another, they form a right-handed configuration, right-handed helix. So when we're talking about their configuration, individually it will be left-handed, and when we talk about when we talk about as a collagen fiber, that is a tropocollagen, then it will be right-handed. Okay, so let me just write down. So when it will be in tropocollagen, that means all the helix will be present together, three helix will be present together, then it will be right-handed. Okay, so as you can see over here that we discussed about the structure of fibrous protein, that is collagen, and it is present. Where is it present? Its length, its diameter, right? Diameter is this particular thing, right? So its diameter, how many residues of amino acid is present in one polypeptide, and then what kind of configuration does the structure has. Now we'll talk about another thing, that is amino acid sequence. So in collagen, the amino acid sequence is present in such a manner that every third amino acid will be glycine. That means every third amino acid will be glycine. So as you can see over here, the amino acid sequence is usually GPX. That means G is for glycine, P is for proline, and then X is for, it could be either hydroxyproline or it could be either hydroxylysine, right? And as I said, every third amino acid will be glycine. It'll be something like this. Let me just write. So it will be G, and then P, and then X, and then again G, and then P, and then X. Okay, so it is not mandatory that this and this will be repeating repeating like glycine, but then it is mandatory that glycine will be repeating in every third amino acid. That means after every third amino acid, glycine will be present. So therefore, it is a kind of like an important factor that decides the structure of the collagen, right? Now, you must be wondering what exactly is this hydroxyproline, right? So hydroxyproline or hydroxylysine. So if you just see the structure, I'm just going to show you the structure. So this is the structure of a proline, right? So we were talking about specifically about proline, and then we said this X could be either proline or it could be lysine, and then we added the word hydroxy, right? So this is actually the structure of proline. And then if in the fourth position, an enzyme called prolyl 4-hydroxylase adds an hydroxyl group, then this will be termed as hydroxyproline, right? And similarly is in the case of lysine as well. This is the structure of lysine. If I just take out the OH, this is the structure of lysine. And if, in the fifth position, if lysyl hydroxylase adds a hydroxyl group, then this will be termed as lysyl 5 then it will be termed as hydroxylysine, right? And then we just have to remember that whenever in the fourth position an hydroxyl group is added, then it will be known as hydroxyproline. And in the fifth position, if in lysine, if hydroxyl group is added, then it will be known as hydroxylysine. But then we have to know the name of the enzyme as well, that is prolyl 4-hydroxylase, because it is it is adding hydroxyl group in the fourth position, that's why. And then it's the enzyme, so hydroxylase. And then similar with this one, so we just have to remember the fourth and fifth position, right? And then one of the most important factor which acts as a cofactor to add the hydroxyl group to this particular amino acid is actually vitamin C. That means vitamin C is a very important cofactor which is required, and then which allows actually the addition of the hydroxyl group to the amino acid. Okay, so I hope you're clear about it. Okay, so now we're going to see so we are sure about the amino acid sequence GPX, right? But the case of GPX is always not there, as I told you. Sometimes the amino acid can have a different sequence as well, but glycine will be the third amino acid anyhow. Now let us see another point, that is 2 alpha 1 and 1 alpha 2. What does this mean? Just a moment, I think I've drawn structure over here. Yeah, so I'm just going to take it up. I'm not going to copy it. I'm not going to cut it, I'm just going to copy it. So let's just see this diagram. I'm going to rub the N terminal and then the C terminal, right? So as you can see that there are 3 polypeptides which is actually making a triple helical structure, right? Now 2 of the, for example, I'll just take blue. This would be alpha 1, right? And then this would be alpha 2, right? And then suppose the pink one is alpha 1 again. So 2 chains of alpha 1 and then 1 chain of alpha 2. I'm just going to write it like this so that it will be easier for you to understand. Just a moment. Okay, so this will be alpha 2 and then this would be alpha 1 and this would also be alpha 1, suppose. Let's just suppose it, and I just want to use, so this would be why do you think there is a difference between these polypeptide? Because there is a difference in the sequence of amino acid. So when we talk about alpha 1 chain, the Normal amino acid sequence would be usually GPX, and then when we talk about the alpha 2, then it could be GXP. That means glycine, then hydroxyproline or hydroxylysine, and then at last proline, right? So this is actually the structure of amino acid, sorry, the collagen, right? So you must be now, you must have understood why have I taken alpha 1, 2 alpha 1 and 1 alpha 2, right? Now this is also known as the beta helix. Now if you go to my previous video about alpha helix, helical structure found in amino acid, you'll be able to understand why this is called as a beta helix. The reason why is because the helical structure contains 3 amino acid per turn, therefore it is known as 3. And then in the subscript we have the presence of 10 atoms, right? And then I've just written out here that the first amino acid which will be participating in the formation of the hydrogen bonds will be the first amino acid, and this would be the fourth amino acid, right? Because N, then we'll have N plus 1, then we'll have N plus 2, and then we'll have N plus 3, right? So which amino acid am I talking about? So 1, 2, 3, 4. So fourth amino acid, right? So if you are still confused, I request you to go back to my alpha helix video and then watch it and come back so that you'll be able to understand what exactly do I mean. But you just have to remember that the helical structure is actually a beta helix, and then beta helix is usually the 310, which it is also known as 310 helix, right? Now let us see another thing. So okay, so as I told about this one also, hydroxyproline as well. So we're going to add an hydroxyl group to either the 4th position or to the 5th position, right? When it was in terms of proline, we added in the 4th position, and in terms of lysine, we'll add in the 5th position, right? Now we have to understand, how many types of bonds are usually present in collagen. Like that means what are the major types of like attraction or forces that usually stabilize this complex structure of collagen. The first one is the steric hindrance of pyrrolidine ring. So let me tell you, this structure is actually known as the pyrrolidine ring, because this is a pyroline structure, right? And then as you can see over here, sorry, this is a proline I'm talking pyrroline. It's a proline structure, and then the ring which is present is in pyrrolidine, right? Now this is actually the pyrrolidine, and then now how do you think like, just a moment, I'll just show you. So I'll just rub it off because we don't need this. Suppose, just let's take an example of this helix. This is also an helix, right? And this is a beta helix. So let's just assume that we have proline over here, right? So let's just say we have proline, and then we have proline over here, and then we have proline over here, and then we have proline over here, and then like let's just continue with proline, right? And then I'll just try to draw the pyrrolidine ring. So this is the proline and this is the pyrrolidine ring. So the pyrrolidine ring is it is coming out of the main, so this is actually the backbone, right? This is the backbone of the alpha helix, but then the pyrrolidine ring is present outside. That means it is extending outwards of the the backbone chain, right? So let's just see, this is also, so now what will happen is, this particular pyrrolidine ring structure will try to break this helix, and then it will try to break in this direction, right? And then this will try to break in this direction. That means like similarly in this direction, and then let's say in this direction, right? But then somehow they are moving towards, like, in the opposite direction. So when they're trying to move in the opposite direction, a force kind of cancels them out. That means if this is moving in this direction and if this is moving in this direction, obviously the force of attraction is going to get cancelled, right? And then the structure of the beta helix will be stabilized. I hope you're able to understand. Let's just take an example of a simple example. If you're holding hand with your sibling, right? If you're just if you are this person and if you're holding hand with your siblings, and then if you have another sibling, sorry, if you have like this sibling, right? So if you're like pulling each other, if this person is pulling in this direction and if this person is pulling in opposite direction, the force actually cancels one another, and you would be able to stabilize yourself, right? You'll be able to like stabilize yourself. I hope you're able to like imagine what I'm trying to say. So you know the same thing happens in pyrrolidine ring as well. So the pyrrolidine ring in the proline will try to cancel one another by moving towards opposite direction, therefore not actually knowing that they are actually stabilizing the beta helical structure, right? So I hope you're able to understand what I'm trying to say. So this is actually the pyrrolidine ring that we talked about. So this is the pyrrolidine ring, and this is like a big chunky structure, and then it's gonna go in the opposite direction, and then the same COOH will be there, and then you'll have a ring structure as well, and this will go in this direction, and then finally it'll cancel out, right? Now let us try to see the other thing. So now the other thing is about the hydrogen bond, right? So steric hindrance of the pyrrolidine ring was one particular factor. Another one is hydrogen bond. We definitely know that hydrogen bond is one of the most important, factors that stabilize any kind of protein structure, right? So suppose if we have, like, I'll just try to draw it over here. Suppose if we have a structure over here, right? And then if we have another structure over here, so suppose we have a glycine, right? And then we have any other amino acid. I'm just going to give like X name, okay? So let's write it down, X, right? So let's say that this particular glycine has an NH bond, right? So it has an NH bond, right? And this particular molecule could have a C double bond O, right? And then now what will happen is, there will be formation of hydrogen bond over the, this is one beta helix, and then this is another one. So this is first beta helix, and then this is second beta helix, right? And then there will be third one as well. So as you would be able to understand that glycine will always have its nitrogen, its amino group participating in the formation of a hydrogen bond, and an X molecule of any other any other beta helix will be participating in the hydrogen bond formation with the carboxyl group. That also you have to remember. So in just general, if I have to like draw, then it would be something like this, like this, like this, right? Because three of the beta helix is like winding with one another, and then there will be a hydrogen bond over here, right? So hydrogen bond over here, hydrogen bond, maybe hydrogen bond, hydrogen bond, hydrogen. So I'm just trying to say that there is a force of hydrogen bond as well that really stabilize the collagen structure. And then let us see the last one, that is the van der Waals force. So van der Waals force is one of the weakest force, and then we usually do not consider, we usually do not think that it is that much contributing to the like stabilizing structure of collagen, but still we'll try to understand it. So as you can see over here that this is actually a helix, right? This is what helix? This is beta helix, and then this is left handed, right? This is left handed. Once they come together, then only we'll call it as right handed, right? So okay, so let's just see over here. So as you can see over here that leucine, every leucine which is present in a heptad manner, that means in a heptad manner means like it is repeating every after every seventh amino acid. That means this is the seventh one, then this is the 14th, then 24th. So as you can see that it is, you know, it is increasing or it is repeating after itself at a heptad manner, right? So this leucine will try to form bond with one another. So one more, obviously one more collagen molecule, collagen like polypeptide will be there, but I've just shown you two. So you know every leucine will be you know try to form a van der Waal attraction with one another, right? And then due to which, you know, there is also like a small bit of attraction between this particular polypeptides, but then not very significant in terms of stabilizing the whole molecule, right? So this was the you know the structure of collagen and all these stuffs. So we're going to learn about you know the proper formation of collagen, like how does it how does collagen forms, and then how does it comes out as an extracellular you know tropocollagen. Then you know some galactose glucose will be added, so that we're going to look in our next video, and I'm going to post it soon. So today we learned about the structure of collagen. It is fibrous, rod shaped. It is present in skin, bone, tendon, cartilage, and teeth. And then the length, diameter, how many amino acids are there in one polypeptide chain, what is the configuration as a single polypeptide, and then as you know as it makes a triple helical structure, what is the conformation like over here, right? And then the amino acid sequence, we learned about this as well. And we know that X could be either hydroxyproline or hydroxylysine. Let me tell you this, that you know hydroxyproline is usually favored rather than hydroxylysine. So GPX is the most common form of amino acid sequence, you know, because I told you that sometimes what happens is GXP is also there might be a possibility that GXP is also present, right? But then the most common one is the GPX. So there are two alpha 1 chain, one alpha 2 chain, right? And it is differentiated in the basis of the amino acid sequence. And then this particular polypeptide is a beta helix. That means you know the first atom and the fourth atom will be able to form hydrogen bonds with one another. If you don't know, please go back to my helix video and then come back. Then we have studied the structure of proline and then lysine as well, right? So proline has a pyrrolidine ring, you know, and this ring is actually a helix breaker, you know, but this ring is also present in beta helix and it forms collagen, and it is responsible for the steric hindrance, which actually you know stabilizes the collagen molecule as well, right? So this is also this is the enzyme which adds hydroxyl group to the proline. This is the enzyme which adds hydroxyl group to the lysine in the 5th position, and this is in the 4th position, right? And then we have bonds that usually stabilize the collagen, that is the steric hindrance of pyrrolidine ring, as I talked about this and this structure. And then hydrogen bonds. Hydrogen bond is usually between the amino group of the glycine and then carboxyl group of any other amino acid. And also remember, the hydrogen bond formed is usually intermolecular hydrogen bond. That means it is being formed between two beta helix. If hydrogen bond was formed between this polypeptide chain and between this atom and this atom, then it would be termed as intramolecular hydrogen bond. But this is forming between a different hydrogen, different polypeptide chain and a different polypeptide chain. Therefore, it is intermolecular hydrogen bond, right? And then we also learned about the van der Waals force between leucine, which was present in a heptad manner, and then the force was usually the van der Waals force, which is usually very very weak, but still it causes a slight bit of attraction in the collagen molecule, right? And then why am I counting from this direction? Why not from this direction? Because whenever a protein is synthesized, the first part synthesized is actually the N terminal, so I'm going to count it from this side. Okay, so I hope you're clear about this video. If you like this video, please like, share and subscribe, and thank you so much for watching.